Hello and welcome back to this class where we are talking about the differential technique, the DPV, and as we have discussed that we can have the different types of modifications in terms of the electrode modifications that we will discuss just now, and before that we can have the development from the electronics part to generate the pulses and the computers what you can have to record those pulses as well as to deliver those pulses to the electrodes. So, what are the basic advantages of this particular technique that we can see that in DPV we get a differential curve. So, is not that a typical staircase light of curve, but we get typical like your absorption spectrophotometry type of curve, which is a differential curve consisting of a peak. So, if you have a maxima or if you have a peak, we can find that corresponding peak as the corresponding potential for your electron transfer reaction. And one more interesting thing is that it is a very sharp peak and the height of which is directly proportional to the concentration. So, if we know the concentration of the analyte in the electrochemical cell and the electrode is dipped in it, and the DPV technique is very much similar to that of your cyclic voltammetric technique, because you do not need to require any kind of special arrangement for your electrodes, because the same electrode what we can use for your cyclic voltammetric measurement can also be used for your differential pulse voltammetric measurements as well. So, for a reversible reaction, the peak potential is approximately equal to the standard potential of the half reaction, because in case of cyclic voltammetric measurements what we have seen that we can have two potential, one is EPA, another is EPC. EPA is for anodic peak potential and EPC is the cathodic peak potential, an average of two which give you the corresponding standard potential for the half reaction if it is for any electron transfer for the complete cycle for oxidation as well as reduction. But why we should go for DPV compared to CV is that it has a advantage, because the individual peak maxima can be observed and substances with half-wave potentials differing as little as 40 to 50 millivolts. So, resolution is very important. So, just now what we are talking about, that if we have these two plots in the cyclic voltammetric one particular way of scanning. So, when we go for, so this is the one part, basically the reverse is also there. So, if it is for CV, so DPV for that will give you from baseline very sharp and prominent one for these two electron transfer reactions, because this separation it can be simply say 40 to 50 millivolt. So, the resolution is very high and we can differentiate out these two in case of DPV. So, from CV we can go for DPV when they have the corresponding overlapping peaks in the range of 0.04 to 0.05 volt, which can be resolved nicely. So, it increases also the sensitivity of the voltammetry. So, you have the linear scan voltammetry, you have polarography, you have then cyclic voltammetry, but when it comes to the question of running DPV, it increases the sensitivity, because it is sensitive and the current height is also little bit bigger than that of your current height what we get for your cyclic voltammetric measurements. And what about the detection? Since sensitivity is high, the detection limit is also very high. So, 2 to 3 orders of magnitude. You see, when we mostly do for your cyclic voltammetric measurements, we start from 10 to the minus 3 molar solution for the basic one, then we go down 10 times dilution to 10 to the minus 4 to 10 to the minus 5 molar solution. But in case of DPV, compared to the classical voltammetry that lies within a range of 10 to the minus 7 to 10 to the minus 8. So, that basically gives a 2 to 3 orders of magnitude compared to 10 to the minus 5 molar concentration what we use routinely for our cyclic voltammetric measurements. So, this just now what we have drawn, so is taken from textbook also, again the Skoog's book. So, that book also shows that how you plot that. So, it is not your I, it is delta I, that is why it is a differential technique. So, differential pulse voltammetric technique, because you plot not I against E, but delta I against E. So, this is the range. So, from 0 to positive magnitude of this, and you measure this as the peak, and this peak is giving rise to the corresponding potential value for your electron transfer for any oxidation or reduction. And the current scale is very less, is in nanoamperes now. So, what you see that you can remember that is also nicely that when we are handling a solution which is in the range of 10 to the minus 7 to 10 to the minus 8 molar solution, but we can handle the current also which is in the nanoampere range. So, that is why these techniques will be very much useful for developing some sensors and biochemical sensors also, and with certain kind of modification in the electrodes. So, what we find that before going to that modification of those electrodes, another technique we briefly notice or briefly know that what is called the stripping methods of electrochemical analysis. So, the stripping method of electrochemical analysis, the name itself tells us that you can go for certain amount of your electrode deposition, and that particular sample can be removed from that electrode surface by a method which is called stripping. So, stripping method is that an analyte you have in the solution. So, you take the analyte in the electrochemical cell and is deposited on a working electrode. So, basically we know that from the very beginning that when a iron nail is inserted in a copper sulphate solution, copper is deposited on the electrode surface. Now, what you do, that analyte can be allowed to go for deposition on the electrode surface by passing electricity. So, if you allow some time, because the deposition will take some time, and after some time the electrolysis is discontinued. So, you stop the electrolysis once you pass the electricity for some time and allow that electrolyte to be deposited either through oxidation or reduction. Then you stirring is stopped. So, when you stop the stirring and the deposited analyte is then determined. So, you know how much analyte is deposited to one of the voltammetric procedures what you have used so far. So, the first of this kind is your anodic stripping. So, you will have definitely therefore, the two types of stripping. What will be stripping method, one will be anodic, other will be cathodic, and is related to your electrode deposition. So, that electrode deposition will be useful for measuring this. When you go for anodic stripping, what we find here, the working electrode is a cathode, that is why we get the corresponding one. So, working electrode is the cathode during the deposition step and as an anode. So, the nature of the working electrode we are changing. When we talk in terms of anodic stripping, so initially you see that once it is oxidized, it is deposited, say, and once it is reduced, it can be deposited. So, you have to go for the reverse process. So, when your working electrode is cathode, you get the deposition, then you change this potential to anode. So, when you go for its nature as the anode, you get the corresponding stripping process, when the analyte is oxidized back to its original form. So, you can have the metal ion. So, suppose it is copper 2 plus, and copper 2 plus can be deposited as copper 0. So, what is that? So, you know that the corresponding one is reduction. So, you require a cathode for that reduction, then you change the nature of that electrode as anode. This will go to copper 2 plus once again. So, when it is the removal of this thing, it is your anodic stripping. So, the process will therefore be known as your corresponding anodic stripping, and the nature, because we are talking so far, we should also be very much careful, forget about your reference electrode, but you have the working electrode and the countered electrode. So, the working electrode is your electrode on which is all these things are happening. So, then the other process is the opposite to the first one. So, when we have the working electrode behaving as an anode, we get the deposition. So, you get that particular deposition and it will be cathode during stripping. So, the process what is known as the cathodic stripping means, because during it is coming out from the electrode surface, it will be released from the electrode surface such that what we have seen that it will be released from as the copper 0 from the electrode surface, because this deposition is there on the electrode surface as copper 0, and when it is oxidized as a part of your process known as anodic stripping process. So, the anodic stripping process will give rise to the corresponding generation of copper 2 plus from copper 0, and it will be removed from the electrode. So, the opposite one will be your cathodic stripping back. Then during the pre-concentration step, that means if we want to increase the concentration by certain step where we know that the analyte concentration is very less or very small, say 10 to the minus 5 molar or 10 to the minus 7 molar. So, if we want to increase the concentration of these things, which is known as the pre-concentration step, the stripping method is the lowest detection limits of all voltammetric procedures, because from the solution you go for deposition as a pure form of the material what you are going to get as deposited form of, say, what the example we got is your, say, copper 0. So, the anodic stripping with pulse voltammetry. So, once you get this anodic stripping with pulse voltammetry can reach to a nanomolar detection limit, because already we have seen that we can detect a nanoampere concentration. Similarly, the nanomolar detection is also possible with some very important metal ions which we know that are present in environment from some automobile exhaust, from some other industrial exhaust or some other processes or activities what human being are doing. So, these three heavy metal ions, say lead, bivalent calcium and monovalent thallium. So, they are very important in environment, and the presence of those in the environment is also very much problematic. So, if we want to detect the concentration of these in very low concentration, because in the environment you do not get a molar concentration or decimolar concentration or a millimolar concentration, because it will go beyond a micromolar solution and it will reach to a nanomolar concentration in the range of 10 to the power minus 9 molar solution. So, these things, so since these are all metal ions, so these stripping processes are very much useful for handling metal ions like copper. As I give you the example, only thing you have to know the corresponding reduction potential values, and it should be achievable through your cell as well as your electrode. So, what we find that if you have a mixture of those two also, just now what we have seen for your copper, so it can be a mixture of copper and cadmium also. So, you have the corresponding time axis, and when we go for these, that means your excitation signal, as we know that how much time you require, so is the time axis you can have. So, when we go for deposition, so you get the deposition, so you require some excitation signal. So, for the stripping process we consider these two as the corresponding excitation signal for stripping determination of cadmium 2 or copper 2. So, what you find, therefore, we can analyze it also that this much time is, so this much time you have to spend, because you have to wait for the electrolysis for your reduction. So, both bivalent cadmium and copper can be reduced. So, the potential of your choice is such that at this particular potential of minus 1 volt, basically both the two species, that means both cadmium as well as copper, can be reduced. So, the exhaustive electrolysis for that particular purpose is that the entire amount of copper or entire amount of cadmium will be deposited on the electrode surface when you apply a potential of minus 1.0 volt. So, at this point, that means if you relax some time, so after some time, because you have to definitely give to some time, because as you switch on the potential to the electrode surface, it will not that all the solution will be deposited as the metal of 0 state of cadmium and copper on the electrode surface, because it is everything is diffusion control, because the entire thing was there in the bulk that has to be moved on the electrode surface. So, after giving some time to stay for your reduction, so you go for that. So, when you reach over there, then if you go for that, so this is the thing, that means the potential you give and you allow for some time where all will be converted to M, is not exactly, we write it as M0, is a nicer way to write as M0. So, this M is the metallic state, then you go for this one. So, if you go for, because this was your cathode, and then you go for the corresponding anode. So, anode means that it will just go for the corresponding potential value, that means you can scan the potential also, because the corresponding thing what we are talking about, this is your differential pulse technique. So, on the electrode surface you are getting stripping, as well as you are trying to record the corresponding voltammogram due to the response of their oxidation of the species like cadmium 0 and copper 0, which were deposited on the electrode surface. So, as you move from, this is -1. So, again we move back, -1 to 0 will scan, and in this particular case your electrode is now anode. So, this anodic electrode now go for your corresponding record of this voltammogram. So, you excite it, then you record it, that voltammogram after this time. So, you allow, this is a very quick process, you do not require much time to spend. So, you get what, that is again a DPV plot, but we now call it as a stripping DPV, because everything is stripped off from the electrode surface, and what we are looking for is not a change between two oxidation states of copper 1 to copper 2 or iron 2 to iron 3, but it is from the state where we are talking about the metallic deposition of copper as well as the cadmium. So, these are the stripping DPV plot. So, the differential voltammogram plot will be, the first cadmium will be oxidized back to cadmium 2, because your potential for this is higher compared to your copper. So, copper will be in this particular range below -0.2 volt, you get the copper. So, with these very distinct and very well established potential values for the oxidation of these things. So, if you have certain amount of impurity in the solution also, that is also a very good technique, because you do not bother about the corresponding impurities, only selectively the ions which are the bivalent copper as well as bivalent cadmium will be deposited on the electrode surface. And you, one thing that is also known as your pre-concentration, because the pure form of cadmium and copper are deposited on the electrode surface, and when you get those pure forms in some solution, it can be your polluted water sample, it can be any other industrial effluent sample. So, those things are known as the corresponding pure amount of copper and cadmium present in that solution, because you are showing some electroactivity on the deposited pure form of copper as well as cadmium. So, the height of these two peaks, as we discussed earlier in case of only DPV, that the heights of these two peaks are proportional to the weights of the deposited metal, because it is related to the concentration, and that concentration is directly proportional to the amount of deposition during the cathodic cycle. So, now we will just simply move to development of some sensors, and these are the typical part where we talk about the application of your electrochemical methods of analysis. So, two things are there, that means when we try to develop a sensor, so the development of the sensor will have something, that means you can have the corresponding thing, that means the electrodes. So, you can go for the modification of the electrodes. So, we get that as the electrodes and their modifications, therefore. So, modification and this thing, how we can develop those things, that means some amount of modification, covering and some other types of activities what you can do on the electrode surface. So, once we change the surface or the nature of the surface, so nature of the surface of the electrodes. So, we basically change those, and as a result we consider some electrodes, which is a very common terminology for those, are known as your surface modified electrodes. So, we get surface modified electrodes. So, pictorially we can identify these, because what are the things we should do, that we should do something on the electrodes for their modification and the selectivity what we can increase. So, if we try to increase the selectivity, so increase of selectivity, how we can do that, how we can increase the selectivity of these species. So, these we can do by attaching some membrane. You know that why we use membrane, because membrane is some kind of filtration process. So, membrane will allow only some species of our choice, of our importance, will pass and will reach to the electrode. So, you increase the corresponding sensitivity. So, the membrane is membrane is one part, and two is your modification of the surface. So, if you have some time, we find that you can have the very overlapping peaks, and we have very close formal potentials. So, having some different electrode kinetics, and you also some adsorption. So, how we increase this particular specificity, because if we want to sense now something which is biological in nature. So, you have the biological analytes. So, how we handle those biological analytes, that we will see. So, this biological analyte means it should have some good selectivity in terms of its corresponding reaction with that of your electrode. So, once we get that modification, and that modification is your on the electrode surface. So, if this is your electrode, so what is this? This is your electrode surface, which will be in contact with the solution containing those analytes, and suppose this gives you the corresponding electron, that means it is responsible for reduction of something. Then we can have something, how we can go for these modifications, and these cyclic processes, all you know now, that will also be very easy to explain with respect to those, because these are all known as your corresponding reversible thing. Two species will be there, X and Y, and they are interconnected through electron transfer reaction. So, the reversibility of these things are also important, because we get this sort of loop for electron transfer reactions as well as in different catalytic cycles. So, to grow, to develop, to establish some electrocatalytic processes also will be dependent on these sort of modifications. So, in the first step on the electrode surface, you can have the mediator. Some simple metal complexes with organic ligands can be a very good mediator, which can immediately be attached, or some other materials like carbon rod or carbon tube or other carbon based materials like that of your electrode, which may or may not be your graphite electrode. So, carbon nanotube can be there, or some graphene sheet can be there. So, these mediators, so once this electrode is going for that, so you get the corresponding mediator in the reduced form, and this can settle between this, that means this radiator when it is going for the oxidized form is MO. Why it is going for your oxidized form? Because your enzyme is also attached to that. So, the mediator is then attached to your enzyme in the oxidized form. That means, your oxidized enzyme will oxidize your reduced mediator, but the reduced mediator we obtain from electron transfer from the electrode surface. So, this will go, so this electrode surface will go like this. So, it is reaction with this Mr. So, E0 plus Mr will produce M0 plus Er, your enzyme in the reduced form. So, as a result what we get, the enzyme in the reduced form. So, that is required enzyme in the reduced form. So, what is that is not in the oxidized form, but enzyme in the reduced form will be utilized to take up your substrate, and when it is reacting with your substrate we get the corresponding products. So, these electron transfer reactions are therefore very much important to know, because these electron transfer can directly go to your reaction with that of our substrate to product conversion, or it can have some idea that it can be mediated by some enzyme. So, that is why we will be handling the biological system. So, some very selective enzymes we can use also. So, an enzyme can be anchored and can be put on the surface of the electrode. So, without the presence of this mediator, you may not have the mediator over here, only that enzyme as well as this thing. So, one thing is you can know nicely that electrode is fine to go for your electron transfer reaction for getting substrate or product. If it is not, then you bring the enzyme. If it is not active in that way for electron transfer directly from the electrode, because this enzyme what we are looking for is reduction from the mediator oxidized to the mediator reduced, is not that it can go for electron for the enzyme directly. That means oxidized enzyme may not be reduced to the reduced enzyme directly by attaching it to the electrode surface. So, that is why you use some mediator. That is why we know there are large number of mediators are available in biological system. Suppose we know that in cytochromes, the cytochrome C oxidase, we know there are large number of mediators are available, and for each and every mediator we have a sequentially the corresponding difference in the corresponding electrode potential values. So, one phase you have the corresponding food material which will be oxidized by O2 on the other side. So, in between you have large number of mediators, that means electron transfer mediators are available, and in some steps only we get the corresponding synthesis of ATP molecules from the ADP molecules if the delta E value rather the corresponding potential difference values that corresponding change in the potential values for these two coupled things are sufficient to go for your synthesis of the ATP molecule from the ATP molecule. So, these mediators are very common thing for all these type of development of the sensors. Particularly we will be discussing two sensors in our next class. One is your oxygen sensor, another is the glucose sensor. So, we can have this that you can bring high sensitivity, therefore, high sensitivity for the corresponding reaction. That means you have to have some biological sensing element, and what we bring the corresponding enzyme, and this enzyme we can put within the membrane, or it can be immobilized on the electrode surface, immobilized on the electrode surface. Okay. So, what we get this biological sensing agent. So, you have the corresponding thing how we can measure this sensitivity. So, you have this high sensitivity we can achieve by putting biological sensing element. So, that biological sensing element is important to know, which can sense the biological molecules. So, it can be immobilized on the electrode surface. So, electrochemical signal what we can get, that we have to monitor either current or the differential current. So, electrochemical signal transduction should be there. Either you record I or delta I for those things, and we can develop in that way the biosensors. So, the electrodes for these sort of measurements, for making or handling the biosamples, oxygen can be there in the biological sample, your glucose can be there in the biological sample. So, when we talk in terms of the corresponding current plot with that of your potential, and these are of two types, one can be your amperometric biosensor, the other can be your voltammetric biosensor. Okay. So, all these things will develop next day, and we will discuss once again all these things for your convenience. Okay. Thank you very much.